Good morning friends, welcome you all on my channel Biomanic. I hope you all are great and having fun. Friends, one request from my side, if you are liking my videos, please subscribe and please press the bell icon because you will be getting the latest updates of my coming videos. Thank you for your support. Today's my topic is the photosynthesis is the main topic and the subtopic is chemosmosis. So okay friends, we'll start with the topic chemosmosis. What exactly the chemosmosis is? In the previous lectures I have discussed that the assimilatory power means the NADPH and the ATP is formed during the photochemical reaction or the light dependent reaction. Now, what is exactly the use of the formation of these assimilatory powers? Why these powers are necessary? As we all know, the thylakoid, the membrane of the thylakoid, there are two types of thylakoids, the granule thylakoid and the stromal thylakoids. The proteins which are present, the enzymes which are present for the formation of food the enzymes which are responsible for the formation of carbohydrates or the food is basically present on the stroma, the matrix. But the photochemical enzymes or the enzymes which helps in the photolysis of water, they steal the electrons from that photolysis reaction, and the electrons with the help of proton carriers is passed with photosystem 2 with photosystem 1, and ultimately it is assimilated with or it is mixed with a proton carrier, an electron carrier, that is the nicotinamide adenine dinucleotide phosphate. So the membrane of the thylakoid and the permeability of the membrane of the thylakoid decides whether the proton can move or facilitate easily or not. So this movement of ions, we can say the movement of protons from the membrane of the thylakoid to the stroma. Why stroma? Because stroma, it is having all the enzymes which are necessary for the reduction of CO2 into carbohydrates. For that reduction, we require what? Hydrogen, as we all know, redox. The reduction is a process in which the substrate, it gains hydrogen. So if the CO2 is to be reduced and to be converted into glucose, we require protons. And these protons are shuttled, these protons are transferred from the lumen to the stroma. Lumen means the space between the space inside the thylakoid. It is also having a matrix, a kind of liquid, and the matrix or the stroma or the liquid present outside the thylakoid. So we have to develop a gradient. Getting my point? We have to develop a gradient, we have to develop a difference, we have to develop a electrochemical potential. And this electrochemical potential is developed by three methods. Okay friends, this is suppose thylakoid, the membrane of the thylakoid, the outer membrane and the inner membrane. This is lumen and this is stroma. So we are having certain types of proton carriers which take the protons, which help the transfer of protons from outside to inside, from stroma to lumen. This is also stroma. This is also stroma. This is also stroma. Okay. So we have to basically decrease the concentration of H plus ions in stroma, and we have to increase the concentration of H plus ions in lumen. Now a question arises, a very common question arises that, sir, why we are doing so? We are doing so because this proton helps, or the gradient of the proton, what we are going to maintain, what we are going to create, will help to increase the quantity of it, will help to make the energy, and that energy is used in the synthesis of ATP. You're getting my point? No. Okay, I will come to this point bilingually. जो H प्लस आइंस हमें स्ट्रोमा में मिल रहे हैं, हमें स्ट्रोमा से उन H प्लस आइंस को अंदर लेके आना है, कहाँ पर? ल्यूमन में, एंड देन वी हैव टू, और अब इस ल्यूमन से हमें फर्दर ट्रांसफर करना है उसको स्ट्रोमा में. तो ऐसा करने पे स्ट्रोमा और ल्यूमन के H प्लस आइंस के बीच में ग्रेडियंट डेवलप होगा, एक डिफरेंस डेवलप होगा, एक टेंशन डेवलप होगी. एंड बिकॉज वी ऑल नो दैट व्हेनेवर the things move from their higher concentration to lower concentration, always and always energy moves from its high potential to low potential. So it releases energy, and that energy is utilized in the form of useful work. As in chemistry we know the Gibbs free energy change. So this energy is basically utilized by ATPase or ATP synthase to convert ADP into ATP. Now, so this membrane side we are having what? Photosystem 1. Photosystem 1, P680. Now the very common reaction is the light, the photons get incidented on this photosystem 1, P680, it gets excited, the accessory pigments, the core molecules, they get excited to emit electrons. The electrons they acquire, or they reach to a higher energy level for a transition time duration of 10 to the power minus 9 seconds. All these electrons are excited with a threshold energy of 23 kilocalories per mole. So these electrons are further excited by P680 to the primary acceptor, that is pheophytin, and after that pheophytin we are having what? The plastoquinone. So this electron, mean the photolysis of water takes place, H2O give rise to 2H plus 2OH minus, 2H2O give rise to 2H plus 2OH minus. Okay, mean we can maintain the gradient of protons by three factors, by the three methods. First is this photolysis of water. Okay, so what is the method to maintain the electrochemical gradient regarding the protons? That is the photolysis of water. We are having protons here, so the protons are increasing in the lumen. Okay, now how can we decrease, or how can we move the protons in the stroma inside the lumen? Okay, so we are having plastoquinone, so the electron and the proton, mean the plastoquinone takes the electron. Okay, we are having 2 mole electrons over here. Okay, so these 2 mole electrons are transferred to what? Plastoquinone. Now plastoquinone we are having H plus over here also. So this plastoquinone basically converts to its reduced form and again converted to plastoquinone. It's a type of redox reaction. Mean these 2 electrons are transferred to PQ, and the H plus taken by this PQ from the stroma to be converted to PQH2, and this PQH2 is further transferred to PQ, mean it is again giving what? H plus आइंस. PQ ने यहाँ पर स्ट्रोमा से प्रोटॉन्स को लिया लाइट रिएक्शन के दौरान जो इलेक्ट्रॉन्स बने थे उन इलेक्ट्रॉन्स को लिया और ये इलेक्ट्रॉन्स और प्रोटॉन्स को कपल करके बेसिकली यहां पर इसके साथ एक साइटोक्रोम B6F कॉम्प्लेक्स जुड़ा हुआ है। तो ये साइटोक्रोम B6F कॉम्प्लेक्स क्या करेगा, PQ को PQH2 में कन्वर्ट करेगा और PQH2 फर्दर प्रोटॉन्स को अंदर दे देगा और वापस से ऑक्सीडाइज हो जाएगा। इलेक्ट्रॉन्स का फ्लो एज सच रहेगा। इलेक्ट्रॉन्स कहां चले गए, साइटोक्रोम B6F कॉम्प्लेक्स में। अब ये साइटोक्रोम B6F कॉम्प्लेक्स फर्दर इलेक्ट्रॉन्स को अलग अलग इलेक्ट्रॉन कैरियर्स के थ्रू, मीन विद द हेल्प ऑफ डिफरेंट इलेक्ट्रॉन कैरियर्स, द इलेक्ट्रॉन्स आर फर्दर पास्ड, दे आर फर्दर ट्रांसफर्ड टू photosystem 1. Photosystem 1 also get excited. So here we are having NADPH, or we can say the NADP plus, which takes these protons from stroma. So we are having NADP reductase enzyme. In the presence of NADP reductase enzyme, the NADP plus or NADPH is converted to NADPH plus H plus. Okay. So these are the three factors which helps to maintain the electrochemical gradient of the protons. The very first is the photolysis of water, which increases the protons in the lumen. Then the transfer of protons from stroma directly inside to the lumen cannot happen, but it can be done with the help of some coupling factors or with the help of some transfer proteins. That is, PQ is converted to PQH2, PQH2 is again converted to PQ. Protons it is delivering inside and the electrons are further transferred. Now with the help of NADP reductase enzyme, NADP plus is converted into NADPH plus H plus. Okay, and we all know PS1 is associated with this coenzyme. Okay, so PS1, PS2 we are having this non-cyclic photophosphorylation. Okay, the non-cyclic photophosphorylation, but we haven't generated ATP yet. So this ATP, we all know during the Z scheme, the cytochrome B6F complex helps in the formation of ATP. Okay, how? Exactly what happens? We are having a coupling factor known as CF0, CF1 particles that we have studied in mitochondria, Fernandez-Morez particles or F0, F1 particles, oxysomes. So these particles help to transfer the protons from outside to inside. As we all know, as the concentration of H plus ions increases, the pH will decrease. The environment becomes more and more acidic. Okay, so we are having like this. This is F0 and this is F1. So the protons which are increased, absorbing protons, giving protons, the protons which are increasing from inside to outside, they are shuttled, and these protons are transferred from higher concentration to lower concentration. So this electrochemical gradient, basically it is an exothermic reaction. It gives off energy and that energy is utilized by ATP synthase or ATPase enzyme, and with the help of this ATPase enzyme, what happens? ADP is forming what? ATP. फिर से समझा देता हूँ। ये जो कपलिंग फैक्टर है, ये कपलिंग फैक्टर बेसिकली जो आपका थाइलाकॉइड होता है, थाइलाकॉइड की मेम्ब्रेन होती है वहाँ पर है, एंड दिस कपलिंग फैक्टर ये हेल्प कर रहा है H+ को अंदर से बाहर लाने के लिए। अब जब H+ अपने हायर कंसंट्रेशन से लोअर कंसंट्रेशन की तरफ आता है तो वो एनर्जी रिलीज करेगा, क्योंकि अभी मैंने बताया कभी भी कोई चीज़ मूव करती है फ्रॉम हायर कंसंट्रेशन टू लोअर कंसंट्रेशन, तो उस कंसंट्रेशन ग्रेडियंट के अलॉन्ग मूव करने के दौरान वो एनर्जी रिलीज करती है, ठीक है ना। तो जब एनर्जी रिलीज होगी तो उस एनर्जी का फायदा कौन उठाएगा, उस एनर्जी का फायदा उठाएगा ATPase, और ये ATPase क्या करेगा, ADP को किस में कन्वर्ट कर देगा, ATP में। इस तरीके से पर 2 प्रोटॉन्स जब का शटल होता है, व्हेन 2 प्रोटॉन्स आर ट्रांसफर्ड फ्रॉम इनसाइड टू आउटसाइड, 1 ATP इज फॉर्म्ड, ठीक है। जब 2 प्रोटॉन्स इनसाइड से आउटसाइड, ल्यूमन से स्ट्रोमा की तरफ आते हैं, तो उस समय एक ATP में कन्वर्ट होता है इन द प्रेजेंस ऑफ ATPase enzyme. So this is all about the chemosmosis and photophosphorylation. Why photophosphorylation? Friends, there are some queries regarding, sir, what are the different types of photophosphorylation? What are the different types of phosphorylation? Phosphorylation means the formation of ATP. If the formation of ATP is taking place in the presence of light, photophosphorylation. If the presence of ATP is taking place inside mitochondria, we can say both in plants and animals, the oxidative phosphorylation takes place. Oxidative phosphorylation means directly in the presence of certain dehydrogenases enzymes or oxidative enzymes, the formation of ATP takes place. So that is what oxidative phosphorylation, because the protons, they are transferred further to oxygen, okay, in the as we have studied in ETS, electron transport system. Here we are having the formation of protons in the presence of what? Light. That is photophosphorylation. And in oxidative phosphorylation we are having one thing more, the substrate level phosphorylation. The substrate level phosphorylation means directly formation of ATP without involving any intermediate carriers like GTP, like NADP, like FMN, like FAD, like that, okay. So this is the overview, friends, regarding the chemosmosis. Now what is exactly the advantage of this chemosmosis? This chemosmosis helps to maintain the osmotic pressure, it helps to maintain the chemical gradient of H+ ions, and these H+ ions are transferred from inside to outside to release energy, and as the energy is packed by ATPase enzyme to form what? ATP. So friends, we will be discussing more details regarding the Calvin cycle, which is my next video. I will be talking about the Calvin cycle in my next video. Till then, bye bye, take care.